So now I am going to discuss about the screening strategies that are normally done just after your creation of your library, your genomic library and cDNA library. So once a DNA library or a cDNA library is being created, the clones, that is your cell lines, okay, that containing your desirable gene fragment, must be screened for your identification of specific clones. Now where your specific desirable clones are, they are in the colony of cells, that can be done only by screening. Screening technique are mostly based on sequence of the clone, which will be complementary base pair by using certain things like probes, primers, etc., and also based on the structure and function of its product. So let us see the different types of screening strategies that are normally employed for identification of your clones obtained from your DNA library or cDNA library. The first method is screening by DNA hybridization. In this case, the target sequence in a DNA can be determined with a DNA probe. The target sequence in a DNA can be determined with a DNA probe. Now, what does DNA probe means? What does DNA probe means? A probe is a single-stranded sequence of DNA or RNA used to search for its complementary sequence in sample genome. So just like a primer, probe is almost the same thing, but there is a little bit different that we will see later on. So probe is a single-stranded sequence made up of DNA or RNA. So here we will use DNA probe so that it can bind our target DNA sequence, and thus it can search for its complementary sequence in the sample genome. Now the probe is placed into contact with the samples under conditions that allows the probe sequence to be hybridized with its complementary sequence. Now the special feature between probe is that probe is often labeled with a radioactive isotope or with a chemical tag, which allows its binding to be visualized. So we can able to visualize them. We can able to visualize the probe, that yes, it is there, that has been complementary base pair with the target sequence. Then this is the difference between probe and a primer. Probe is labeled with a radioactive isotope or with a chemical tag that allows its binding to be visualized. We can visualize them. How you can visualize them? By autoradiography techniques, by other flow cytometry, different type of techniques that can be used, can be employed. In a similar way, labeled antibodies are also used to probe a sample for the presence of a specific protein. Just like in the same way, when you want to visualize your target DNA or RNA sequence, we use a probe, a short DNA or RNA sequence tagged with a chemical or with a radioactive substance so that it can be easily visualized. In case of chemical, we can go for some substrate, some colorless substrate, some chromogenic substrate, which will on upon reaction with the enzyme, it will give some colored product. Then also we can able to visualize them, or some fluorescent substance which will emit light, okay, on treating with certain chemicals. So these type of substance are normally labeled with a probe. So let's come to the topic. So here the target sequence in the DNA can be determined with a DNA probe, And to start with, the double-stranded DNA of your interest has to be unwinded. For that, we can go for denaturation by raising the temperature. That 95 degrees Celsius is enough for the DNA to be getting denatured, or you may go for alkali treatment if you think that heating can cause loss of your base sequence and can cause mismatches or alteration of base. So we can go for alkali treatment for denaturation of DNA. The two DNA strands are kept apart by binding to solid matrix. Now here the solid matrix that we use is nitrocellulose filter. Okay, so nitrocellulose filter are discs like substance which are normally placed on agar plates. Okay, and your DNA of interests are actually allowed by, after denaturation, they are allowed to bind to that solid matrix like your nitrocellulose or nylon membrane. They are highly inflammable membrane. Okay, now the single strand of DNA probe, which is about 100 to 1000 base pair maybe long, they are labeled with isotopes. Radioisotopes are added. That means the single strands of DNA probe of 100 to 1000 base pair long having labeled with radioisotopes are being added so that they can complementary base pair with these DNA strands that we are looking for. Hybridization, that is base pairing, occurs between the complementary nucleotide sequences of the target DNA and the probe. So hybridization is then done, base pairing will be done between the probe and their complementary sequence where it is located, if they are located. For stable pairing, at least 80% of your base between the two strands should be matched, otherwise there will be no proper base. At least 80% pairing should be done between the base with the probe. And finally, after binding the hybridized probe, the unhybridized probes are washed away and by certain buffer solution, and then the hybridized DNA can be detected by your autoradiography. So what is autoradiography? It's just like a photographic film. Autoradiography is the bioanalytical technique used to visualize the distribution of your radioactive level substance with radioisotope in a biological sample. So we are using the probe which is tagged with a radioactive substance, and with the help of autoradiography you can able to visualize that where that probe is located. It is a method in which radioactive material can be localized with a particular tissue, with a particular cell, organelle, or even biomolecules. It is very sensitive technique and being used for various biological experiments. The main principle of this autoradiography is this, that autoradiography is based upon the ability of a radioactive substance to expose the photographic film by ionizing it. So if you use the radioactive labeled probe, and if you use a autoradiographic film, which is a photographic film normally that we use, that radioactive substance if present, they will as they get exposed to that photographic film, they will try to ionize it, because they emit continuously they are emitting the beta particles, alpha particles, right? In this technique, a radioactive substance is put in direct contact with a thick layer of photographic emulsion having a thickness of 5 to 50 millimeter. Now that photographic emulsion have gelatin like substance and silver halide crystals, and this emulsion differs from the standard photographic film in terms of having high ratio of silver halide to gelatin ratio. Now this photographic film is quite different from the normal photographic film because they have a high ratio of silver halide to gelatin. That means the quantity of silver halide is quite greater than gelatin in this photographic film, and small size of the grain. And then it is left dark for several days for proper exposure, so that DNA sample which has the probe has been hybridized, when we have exposed them with photographic film, it will emit the particles and it left them in the dark for proper exposure. Silver halide crystals are exposed to that radiation emitted from that probe, which chemically convert that silver halide into metallic silver. That gives you a dark color band, and the resulting radiography can be viewed through electron microscope, through pre-flashed screen, through electrophoresis, through digital scanners, etc. So normally an autoradiograph that we prepare is just like an X-ray film or a nuclear emulsion produced by patterns of decay emissions. Okay, that is your beta particles or gamma rays that emitted from the radioactive substance exposed to the film and reduce that silver halide present in the photographic film into metallic silver, which gives you a dark color band that can be detected further by that plate. So in this way we can able to do this DNA hybridization simply for identifying our target sequence, that where our target sequence is located in which cell. So simple thing, you can see in the figure the source DNA double stranded. We make them denaturation by alkali treatment or by increasing the temperature. Then we allow that DNA, single stranded DNA, to be bind with the membrane so that they can bind with solid matrix like nitrocellulose or nylon membrane. Then for hybridization we use here single stranded labeled DNA probes, and you can see that the radioactive label probes is being used. They are allowed to hybridize, and if there is a complementary sequence for that probe, they will bind. The non-bound probes are washed away by a suitable buffer solution, and finally they are visualized in autoradiography. So this is a simple procedure for your detection of your particular DNA. Next is about the DNA probes. They are saying that DNA probes used for screening purpose can be synthesized in many ways. Now you can prepare DNA probes in variety of ways. Let us see. This is not a technique, this is actually preparation of DNA probes. The first one was the technique, that is your DNA hybridization. Here they are talking about that how you can make DNA probe. Let us see. Then again we will get a screening process. So what is DNA probe? Let us see. So DNA probes can be used for screening purpose, can be synthesized in many ways. Let us see. Random primer method. The first method that we go for is random primer method. In this way we can prepare DNA probes just like a primer, but tagged with a certain enzyme or radioactive substance or some fluorescent substance. So in random primer method, radioisotope level DNA primers can be produced by this technique. The double stranded DNA containing the sequence needed to serve as a probe is denatured. Again I repeat, the double stranded DNA containing the sequence needed to serve as a probe is denatured first. A mixture of synthetic oligonucleotides with all possible combinations of base are being given. With a length of 6 nucleotides, each serve as a primer. So at least 6 nucleotides should be elongated, and out of these 4 possible bases, one base has been tagged with a radioisotope. So a mixture of synthetic oligonucleotides with all possible combinations of bases with length of 6 nucleotides each serve as a primer. Some of this primer will complementary sequence will hybridize with the template DNA. The occurrence is entirely by chance actually, because we are allowing these bases to be tagged with a DNA with a radioactive isotope, and we will try to elongate this primer, this probe, as done by polymerase, including the base that has been tagged with a radioisotope. So by addition of 4 deoxyribonucleotides, one of them is radio labeled, as I have said. Maybe you have radio labeled your adenine, or all the guanines has been radio labeled. So they will be included in the DNA chain during elongation process catalyzed by the polymerase enzyme, just like we have done in PCR. So it is purely by chance, that's why they have said. So by the addition of 4 deoxyribonucleotides, one of the nucleotide has been labeled with radioactive isotope, and in the presence of the enzyme called DNA polymerase of E. coli, which is the Klenau fragment has been used here. The Klenau fragment is the large subunit of DNA polymerase of E. coli. The primers are extended on the template DNA, and since radiolabel is used, the newly synthesized DNA fragments will have some radioactive substance as appropriate bases, and they are used now as DNA probes. So in this, the probes are being prepared. A number of DNA probes can be produced from an unlabeled template DNA. Now sometimes non-isotopic DNA probes are being used because radio-labeled isotopes have some toxic effects. It also causes some environmental pollution also. So nowadays non-isotopic DNA probes are also being used, like by using the same method, by using the same primer extension method as described above, as I have said. Sorry for that. By using the same method like the primer extension method, we can also allow to tag that probe with some other materials which can be easily visualized, like we can go for some fluorescent substances, like we can go for biotin also. Okay, let us see. We can go for epitope binding also. Epitope is actually your antigenic determinant. It is the part of an antigen that will be recognized by our immune system cells, specifically by our antibodies produced by the B cells and also by the T cells. So epitope can be also used as a tag to the probe. You can also go for biotin, as has been written in that internet source. Biotin is also called vitamin H or vitamin B7 or vitamin B8. It is a water soluble vitamin, and it is involved in wide range of process. So here we can use this biotin as a tag, as a source to detect, and also we can go for some enzymatic procedure. Some enzymatic can be used. Some enzymes can be used to tag them, like horseradish peroxidase. Okay, alkaline phosphatase can also be used, as we will discuss them later on. Next is screening by colony hybridization. So the above was how to make probe, and we have discussed one of the screening strategy, that was DNA hybridization. The next is your colony hybridization. The DNA sequence in the transformed colonies, that means those colonies where the cells have been transformed by your vector containing your DNA of interest, it can be detected by hybridization with radioactive DNA probe. Sometimes labeled RNA probes can also be used. Colony hybridization technique, also called as replica plating, the same replica plating that we have done in PBR322 experiment to find out that which cells have your desired recombinant PBR322 containing your DNA. Same procedure here, it is also being done. A bit of modification is made here. Let us see the technique. The transformed cells, that means the cells having that vectors containing your desired DNA, grown on colonies on a plate, on a Petri plate known as master plate. Sample of each colony are then transferred to a solid matrix, and that solid matrix is your nitrocellulose or nylon membrane. We just try to press that nitrocellulose or nylon membrane into that master plate so that some part of the colony may be stick into that membrane. That is called replica plating. Now we will transfer into a new, we will transfer them into new Petri dish. Okay, we will transfer that nitrocellulose membrane to a new Petri dish so that we can able to mark the colonies, and also we are allowed to grow that colonies in a new agar plate. The transfer is then carefully carried out to retain the pattern of colonies very carefully, that we are actually taking the colonies from the master plate via your nitrocellulose membrane. We are transferring them into a new Petri dish, and we will do the reference marking also. Thus nitrocellulose paper containing the photocopy pattern. So we just like a photocopy, we press it with the help of suitable source, we press it and we introduce that nitrocellulose paper into a new fresh prepared Petri plate containing your agar medium, nutrient medium for the bacterial cell to grow. The colony cells are then lysed and deproteinated. Deproteinize means we remove all the protein parts. The DNA is then denatured and irreversibly bound to the matrix as stated in DNA hybridization. As stated in DNA hybridization, the DNA is denatured by alkali treatment and irreversibly bound to the matrix, that means to the nitrocellulose membrane. Now a radioactive DNA probe is added the same way that I have said before in case of DNA hybridization. So a radioactive labeled DNA probe is added, which will hybridize with the complementary target DNA if present. The non-hybridized probe are washed away by suitable buffer solution, and the colony of the hybridized probe can be identified on a autoradiograph film, on a autoradiograph plate better to say, and the cells of the colony from the master plate can be isolated and cultured. We can get the reference point that yes, this was the plate, this was the plate from where this was the colony from where our gene of interest is there. So we can do them as a reference marking, and also we will mark the master plate, the colonies from the master plate, and they are further isolated and cultured. So many times multiple colonies are detected. You can get multiple colonies also on hybridization by a DNA probe, and this occurs due to problems associated with master plating. That means by overlapping sequence also it occurs. To identify that which colony have your complete sequence, data is observed from restriction nucleus analysis will be helpful. So sometimes overlapping colonies may occur from the beginning, that I have mentioned in your DNA library preparation, that we cleave them partially by restriction endonuclease enzyme. So sometimes what happen, overlapping colonies can occur, overlapping sequence can occur, and due to which it can give you a missed result. To overcome this problem, you should observe the data obtained from restriction endonuclease digestion. It will be helpful for detecting them. Now normally there is a modification of this colony hybridization technique. So several improvements, let us see. For before that, let us see the picture that has been given, so it will be easy to understand. So here your colonies are there as shown in circle. This is the master plate. The colonies are transferred from master plate to your nitrocellulose membrane simply by sticking them with the help of wooden cork, okay, containing your nitrocellulose filter membrane. We allow them to touch and press a little bit, so that some of the colonies may get stuck into this nitrocellulose filter membrane. And now we will allow that nitrocellulose to be treated in various ways, like lysing. Lysis means lysis of the cells, so that all the protein contents will be removed out. The DNA content will be denatured by suitable alkali treatment, and also to allow that DNA to stuck to the nitrocellulose membrane. And then we apply probe. Now to stuck for attaching this sequence into the nitrocellulose membrane, we go for some hot treatment, some steam treatment is done, then attachment of probe. Now if the probe has some complementary sequence, that is your target DNA, then it will obviously bind. The probe has been labeled with a radioisotope, and here it shows that it has been attached with one of the colony, one of the reference colony, okay, the hybridized probe. Then washing is done. Washing is done to wash away the unhybridized probe, and then further the colonies are detected. It is detected in autoradiography, and with respect to this, we will also observe the master plate colonies, that yes, this was the colony that have our DNA of our interest. So in this way you can go for colony hybridization, but there are some modifications of this colony hybridization is now being carried out. So several improvement in the colony hybridization technique, which has been described above, has been made in recent years. Like your plaque hybridization is being done nowadays. I will tell you about the plaque lift hybridization after reading this part. So in the plaque lift technique, also called plaque hybridization, the nitrocellulose paper is directly applied on the upper surface of the master plate by making a direct contact. That thing I have already discussed with you. By this way, the plaques can be lifted and several identical DNA prints can be made from a single plate. This technique increases reliability. More recently, screening of DNA libraries is carried out by automated techniques like PCR. So what is plaque hybridization? For that I have to go through the other source. Okay, let us see. So the plaque hybridization, also known as plaque lift, was developed by Benton and Davis in 1977 and employs a filter lift method applied to the phage plaques. Phage plaques are the plaques created by the bacteriophage. Bacteriophage normally lyse the bacterial cells here, and thus it forms plaques. This procedure is successfully applied to the isolation of recombinant phages by nucleic acid hybridization, the same process that I have discussed before. It is the same process that is done for this technique, and probably the most widely accepted method of library screening. The method of screening library by plaque hybridization is described below. So what are the steps? Let us see. The nitrocellulose filter is applied to the upper surface of your agar plate, making a direct contact between the plaques and the filter. The plaques have already been done. The colonies have already been treated with the bacteriophage, okay, as a vector. So we make a direct contact with the nitrocellulose filter paper with that plaque colonies. The plaque contain phage particles. Obviously the plaque contain phage particles as well as considerable amount of unpackaged recombinant DNA which will bind to the filter paper. So the plaque colonies will obviously contain phage particles and also some amount of your considerable amount of recombinant DNA which is unpackaged, and that DNA will bind to that filter because the nitrocellulose filter are meant for binding with the DNA. Then the DNA is denatured usually by alkali treatment and is allowed to fix to the filter firmly, hybridized with a radioactive probe as I have mentioned with you, and they are acid, they are detected by autoradiography. So simple thing, first what we have done, the nitrocellulose filter is applied on the upper surface to the agar plate, making a direct contact with the plaques and filter. Then the plaques containing phage particles as well as considerable amount of recombinant DNA is also found on that plaque colonies, and this recombinant DNA will be allowed to bind to the filter paper. Then the DNA is denatured by suitable alkali treatment. They allowed to fix to the filter paper and hybridized with probes. Here we use radioactive probes again, and then the unhybridized probes are washed away, and then they are assayed by autoradiography. So there is various advantage also. Most of the advantage are observed in this type of method because this method results in a cleaner background and distinct signal, less probe hybridization for lambda plaque screening due to less DNA transfer taken from the bacterial host to the nitrocellulose membrane while lift plating rather than the bacterial colonies. So actually what we have done, as we have lysed them with plaque or lysed them with lambda phage particles, so we are not directly allowing the bacterial cells to come into contact as we have done in colony hybridization, but in plaque hybridization, due to the lysis of the bacterial cells by the phage particles, so some of the DNA are actually get allowed to bind to the nitrocellulose filter membrane rather than the whole colony cells attached to the nitrocellulose filter membrane as we have done in colony hybridization. So this is an advantage. It is a cleaner technique. Multiple screening can be done from the same plate, as plaques can be lifted several times. You can lift the plaques several times, not only one time but many times. You can screen them by making contact with the plaque colonies with the nitrocellulose filter membrane. Screening can be performed at a very high density by screening small plaques. You can screen the small plaques also. High density screening has also advantage that large number of recombinant clones can be screened for the presence of sequence homology. sequence homologous to the probes in a single experiment. So this is the technique that you can see here, the plaque hybridization or the plaque lift. You can see the individual phage plaques, master plate of lambda phage, plaques on E. coli lawn, right? Then we apply nitrocellulose filter placed on that master plate to pick up the phage from each plaque, and then it has not been shown that the plaques has been lifted, but it has been lifted. Along with that, some of the DNA, they get stuck into this nitrocellulose filter membrane. Incubation of the filter to lyse phages and denature release phage DNA. Then single stranded phage DNA bound to the filter. Then the same process as we done in colony hybridization with the label probes. Unhybridized probes are washed away by buffer solution and they are detected in autoradiography plate. Now the signal that appears over the phage DNA, that is complementary to the probe, and thus we can see that this colony phage plaque colony has actually is the place where your DNA of interest is located, and we will detect them further. Okay, so this is the technique of plaque hybridization or plaque lift technique. Then screening by PCR is also being carried out. Let us see. So polymerase chain reaction is a good way as hybridization technique for screening for linear libraries, but we need some information on the flanking sequence of the target DNA. It must be available so that we can prepare primers from them, and that can able to bind with that target region. The colonies are maintained in multi-wall plates, and each well is multi-well plates, sorry, it's multi-wall, not, it will be multi-well plates, and each well is then screened by PCR and the positive wells are identified. So PCR can also be employed for screening purpose, but for that we need some information regarding the flanking region of your target DNA, and that flanking region with the help of this flanking region sequence we can prepare primers for this, and that primers will be tagged with certain radioactive substance or with the help it is bound with certain chromogenic substrate which will bear some colored products. Now screening by immunological assay. Immunological techniques can be used for detection of a protein or a polypeptide. Immunological techniques can be used for detection of a protein or a polypeptide synthesized by that gene. So if your main purpose is to detect your protein, to detect that particular polypeptide synthesized by the target gene through translation, we can go for immunological techniques. The procedure adopted for immunological assay and hybridization techniques are quite comparable. Screening procedure by immunological assay is given in figure 9.8 as you can see and briefly discussed below. So let us see. First we will discuss and then we will go through this diagram. So simple thing, the cells are grown as colonies on the master plate. They are transferred to a solid matrix like nitrocellulose. colonies are then subjected to lysis, and the release proteins bound to the matrix. These proteins are treated with a primary antibody which will specifically bind to the protein. Now here the protein is acting as an antigen, and on the basis of that we will prepare such antibody, and here the antigen actually is that protein which has been encoded by our target DNA. After removing the unbounded antibody by several washings, secondary antibody is added which specifically binds to the primary antibody. So simple thing, the cells are grown on colonies on master plates. Cells are grown as colonies on the master plates which are transferred to a solid matrix like nitrocellulose. Okay, the colonies are then subjected to lysis, and then the proteins that are released, which they are allowed to bound to the matrix, to that matrix, that is your nitrocellulose matrix. These proteins are then treated with primary antibody. Here the proteins are acting as antigens, and specifically we have prepared antibody for that particular protein so that it can bind with that. And after removing, we have to wash with suitable buffer solution to remove the unbounded antibody, and then secondary antibody is then added which is specifically designed to bind with the primary antibody. Again the unbound antibodies are washed away. The secondary antibody carries an enzyme labeled at their constant region. The secondary antibody carries an enzyme, okay, into their constant region. If you look at the structure of antibody, it has a variable region and constant region. So the constant region of this secondary antibody has been labeled with certain enzymes like your horseradish peroxidase or alkaline phosphatase bound to it, and then the detection is so devised that with respect to that particular enzyme, when we give suitable substrate, that enzyme is specific for that substrate obviously, and presence of enzyme in that region, in that nitrocellulose membrane, it means the enzyme has been tagged with bind with the secondary antibody which has been interact with the primary antibody which has been interact with that particular protein. That means if the suitable substrate is given and if there is an enzyme for that substrate, then obviously it will react with that substrate and bears a colored product. The substrate is generally a chromogenic substrate which will bear color. I will give you the examples later on. For the horseradish peroxidase, there are certain chromogenic substrates generally used, and for the alkaline phosphatase are also some substrates which are generally used for bearing color product, and thus you will get a positive result, which are the colored spots that are identified. The cells of a specific colony can be subcultured from the master plate. So in this way you can able to detect your particular protein. Okay, so here is the diagram. You can see a master plate, colony of cells are transferred, then a solid matrix, it is allowed to by master plate, by replica plating we transfer that colonies into the solid matrix. Lysis of the cells, proteins are allowed to bound to the matrix, and then we allowed primary antibody to bind which is specific for that particular protein. Here the protein is acting as antigen here for that antibody. Then secondary antibody is allowed to bind. Now the secondary antibody shown here is like black dot, and the secondary antibody actually has a labeled enzyme like horseradish peroxidase or alkaline phosphatase, which will eventually bind with the primary antibody if present. It is designed in such a way so that it can bind with primary antibody, and thus the secondary antibody, the unbound antibodies, are washed away by buffer solution, so that we get a correct result. And then we add a particular substrate for that particular enzyme which has been bound with secondary antibody, and thus a colored product is observed that will allow for us to detect that which colony having our desired DNA sequence. Okay, so this is the thing of immunological assay, the screening of immunological assay. Then screening by protein function, by on the basis of protein function we can also go for screening. So if the target DNA of the gene library is capable of synthesizing protein, that means we have constructed such gene library. For further expression we have included them in such vector which expresses that protein into a suitable host that is not normally produced by the host cell. The protein activity can be used for screening. So it is, again I repeat, if the target DNA of the gene library is capable of synthesizing protein, maybe an enzyme, which is not normally produced by the host cell. So host cell is not normally producing by it, but they are capable of producing protein. Then the protein activity can be used for screening. A specific substrate is used, and its utilization by colony of cell indicates that the presence of enzyme is acting on the substrate. For this purpose, what we can do, just we can allow a specific substrate to be used here. Now if the enzyme produced by that particular gene is carried out, then that enzyme must react with that specific substrate, and utilization by the colony of cells will be an indication there is a presence of enzyme in that cell, that particular enzyme in that cell, and that means that desirable DNA is present. For instance, the gene coding for enzyme beta galactosidase or alpha amylase can be identified by this technique. How? Same thing by the blue-white screening. Suppose you want to detect the gene in your colony that codes for the enzyme beta glucosidase or galactosidase or alpha amylase. So what we can do, we can allow a particular substrate to be acted upon by it. Suppose the substrate is XGAL, 5-bromo-4-chloro-3-indolyl beta-D galactopyranoside. This is the full form of XGAL, and when it is added to the colony, then what will happen? If the beta galactosidase is present, it will react with that XGAL and form a blue colored product, and that will indicate that this enzyme is active. That indicates also that the gene responsible for that formation of enzyme is there, and in that way we can go for the screening by protein by looking at the protein function actually. So this was the all about the screening techniques, screening by protein function, then by immunological assay we use here we use the proteins also. We are using the proteins here for detection of your gene. Then it has not been given details about the PCR technique. Then the Pluck lift, which is not generally used, which is a modification of the colony hybridization technique, and the colony hybridization that that we have said, and your DNA hybridization. So this is all about the screening.